Welcome back to Think Cosmos, where we ponder the universe's deepest secrets and wonders. Picture the glare of fluorescent lights in a late-night lab. Petri dishes lined up like tiny landing pads. In one dish, researchers place a square of smooth, transparent film. The same medical-grade plastic used in catheters, sutures, and surgical net. They add a cloudy droplet of Pseudomonas aeruginosa, a bacterium infamous for clinging to hospital surfaces and dodging antibiotics. Hours pass. Under the microscope, filaments of polymer begin to pit and fray. 48 hours later, the film is stippled with holes. By day 4, only ragged edges remain. Chemical assays show the microbe has snipped long plastic chains into lactic acid fragments, and then slurped those fragments for fuel. Something no organism had ever been documented doing with this material. The breakthrough lands in nature. A hospital superbug can digest medical-grade plastic. The team traces the trick to two enzymes, PaeLase-A and PaeLase-B, that latch onto ester bonds and slice them with surgical precision. When the genes are knocked out, the bacteria lose their appetite. When the genes are cloned into lab-safe E. coli, the humble gut bug gains a taste for the same polymer. Suddenly, the field sees a toolbox. Gene cassettes that could be dropped into fermentation vats or biodegradable dressings to kickstart plastic breakdown on demand. Yet, the discovery comes with a shiver. Pseudomonas aeruginosa already causes hard-to-treat infections in ventilated patients and burn wards. If it can live off IFU tubing or implant coatings, it might linger longer on life support gear. Infection control teams start mapping biofilm hotspots and debating whether to swap in alternative materials. But zoom out, and a different opportunity emerges. Hospitals worldwide discard thousands of tons of single-use plastic every day. Gloves, syringes, surgical drapes, most contaminated, all difficult to recycle. Enlisting microbes that chew medical plastics into harmless monomers could slash landfill loads, cut incineration emissions, and maybe even funnel the breakdown products back into manufacturing loops. Now lean forward and imagine the year 2080. Inside a domed colony on Mars. Every gram of imported material is precious. Waste streams must become resource streams. The settlement's clinic generates a steady trickle of polymer scraps. Flexible tubing, diagnostic cartridges, microfluidic chips. Instead of shipping refuse back to Earth or burning it in thin Martian air, colonists feed it to a sealed bioreactor, seeded with an engineered, non-pathogenic cousin of today's plastic-eating microbe. Within days, the scraps dissolve, releasing glycolic and lactic acids that downstream microbes ferment into ethanol and then into fresh polyethylene furanoid pellets for 3D printers. Medical waste becomes rover parts, greenhouse trays, or replacement visor seals, closing a vital loop in an environment where supply ships are rare. Extend the vision to asteroid mining camps. Prospectors wrap extracted ore in polymer liners before firing it toward orbital smelters. Once the cargo is unloaded, liners are shredded and sluiced into a biotank where enzyme cocktails, descendants of PaeLase-A and B, reduce them to carbon feedstock for onboard bioplastics printers. Every mission leaves with as much packaging capacity as it arrived with. No extra mass required. There's a life support angle too. Future habitats will rely on mixed microbial consortia that recycle air, water, and solid waste. Plastic fragments shed from suit fabrics and equipment could clog filters or poison algae ponds over decades, embedding slow-release enzyme beads, bio-derived from our hospital microbe, into filtration membranes would let the habitat digest rogue polymers before they accumulate. Of course, safety is paramount. No one wants a pathogen breeding in the ventilation ducts of a lunar base. Synthetic biology groups are already drafting kill switch circuits The microbe thrives only in a broth laced with a Martian common mineral supplement. Remove the supplement and its metabolism stall. Other teams design cell wall tweaks that make the strain susceptible to a harmless sugar analog unavailable in human tissue, ensuring accidental infections can be stopped with a simple syrup rinse. Back on Earth, the same enzymes could line smart bandages that dissolve on command, or coat biodegradable sensors that vanish once their data is logged, turning healthcare plastics from enduring pollutants into ephemeral tools. A single Petri dish observation, Pseudomonas nibbling medical plastic, has opened a channel from intensive care units to off-world cities. It hints that the trash-to-treasure alchemy we'll need for sustainable space colonization might already be hiding in hospital corners, waiting for scientists to notice and re-engineer the feast.